Hello my dear students, I hope all of you are doing very well. My name is Abhijit Devnath. Since last few days, we are actually learning this subject called pharmaceutical quality assurance. We are trying our best to boost up your understanding in pharmaceutical quality assurance, so that you become a very competent, fit person to do or to work in future in pharmaceutical industry, so that you can prepare a very good product. So, continuing our lecture, this is the 19th lecture of our entire learning series. Today, I am going to tell you about the protocols for conduct a non-clinical laboratory studies. I have divided this lecture into two different parts. At first, I will tell you about the protocols, then I will tell you how to conduct a non-clinical laboratory studies. Now, let me start with the protocol. Protocol, the word itself, it is telling that there will be some norms, some rules that you have to follow, some details that you have to follow during throughout your studies. So, what is the protocol? Each study shall have an approved written protocol that clearly indicates the objectives and the methods for the conduct of the study, and the protocol shall contain following information. What are the information? A descriptive title and the statement of the purpose of the studies. For example, you are trying to do some work in to identify the activity of a cancer drug. You are working on some cancer that is a project. You have identified some molecule that is showing proper anti-cancer activities, and you are trying to know that whether your molecule is showing anti-cancer activity or not. Okay. So, in your title that thing should be come, and what purposes you are doing. Your purpose is to see whether your drug is showing anti-cancer activity or not. So, that is why when you are actually making the protocol, actually this protocol is actually coming when you study your design. When, for example, you are trying to do some that is a non-clinical work, at that area ultimately it will be finalized. Then identification of the test and control article by name, chemical, abstract number, also code. Name, for what you are doing, chemical, what kind of chemicals you are actually using, it is a natural or it is synthetic or what its chemical name. But many of you might be thinking that, sir, if I mention the chemical name, and if anybody that is got to know about the chemical name, so if he or she again repeat the experiment in his lab, and if they get proper results and patent it, then what will happen? Then I will lost my complete intellectual property right from this. Answer is yes. That is why in many of the cases we are actually using the code number. If you do not, if you are interested, if you are not interested to write the actual chemical name, which chemical you are actually using, in that case you can use some code number TXR001. Who will be able to know about this? You have, for example, 3 molecules, molecule number 1, molecule number 2, molecule number 3. Okay. For example, molecule 1 is the paracetamol, molecule 2 is ibuprofen, molecule 3 is, for example, amoxicillin. So, you have given some code. For example, let me take the eraser. For example, this is the common code for all, and you have added this TXR001 M2, TXR001 M3. Okay, this is the code. So, such kind of codes you can use. Okay, nobody able to understand that what is the actual substances, but still before sending to various kinds of analytical test like NMR, then your other kinds of spectrophotometric test, you should be very careful, because on those cases if somebody is a very good medicinal chemist, analytical chemist, he will be able to identify that yes, the peak is showing in this range, and there are chances it might be this molecule. Okay. So, there are some possibilities, but still you should use some codes. Again you also should mention, let me erase all those things. You should also mention the name of the sponsor, then the name, addresses of the testing facility at which the study is actually being conducted. Okay. Sponsor name is very much important, because ultimately who is actually sponsoring your this project, who is actually sponsoring your this entire activity, because you see drug discovery is not a simple work, it is a long work and lots of challenges. There are more than 20 to 25 challenges are actually available in drug discovery. In common, if I count all those things, there might be more than 100 challenges in drug development and drug discovery. So, that is why a sponsor will give you the money, they are going to tell you they are going to invest the money in those all these processes. So, that is why you should name the sponsor. There are other activities, other things are also associated. Some sponsors actually only working on the cancerous work. So, it will help to understand if anybody is actually trying to evaluate, for him it will be very easy to see. Then you should also give the number, body weight range, sex, source of supply, species, strain, substrain, and also age of the test system. It is also very much important. The number, how much number it will be actually required, what number of the substance of the test are actually going to use. The body weight range, for example, 5 to 10 kg, I am just starting a number, a range. Okay. Sex, male or female, then source of supply, from where you are actually getting these animals. If you are doing the XML, this study on rat, from where you are actually getting. Then species, what species you are actually using, strain, if you are using some microbial things, then you have to mention the strain, substrain, and also age of the test system. Age will play a very big role, because of the metabolism and also excretion, also age will play a very big role. In excretion, those are old age people, or those are the old age animals. If you are doing some preclinical studies, these old age animals, their nephron is not working properly, their kidney is not working properly. So, excretion rate will be very much low. That is why age is very much important. Also metabolism also it will be play a very big role. In many of the cases it will play a very big role. The procedure for identification of the test system, it should be also there, and a description of the experimental design, including the methods for the control bias. All of you have seen there is something called type 1, type 2. Okay. So, in those cases, if there is some bias, the method of control or the bias, if there is here, so that thing should be also addressed in a proper manner. So, that is why a description of the experimental design, okay, including all the methods should be there. You should give a proper description of them, how the method was designed, if you are doing some factorial and other kinds of study design actually involving. So, that thing should be properly give in a description, because you see if we are doing some study and it has some that is a impact on the environment, or it is showing some biological or other kinds of it is creating some biological other kinds of hazards. In that scenario, what will happen? In that scenario, there will be some problem from the people who are living on those areas, because it will obviously it will create some issue. So, at that time the health officials will visit in your testing facility, they will ask to show them the proper the documentation, and there if they see the description, the experimental design, they will be satisfied that yes, the work is going properly, but there might have some mishap may happen. You have seen in the story of recently outbreak COVID-19, SARS-CoV-2. There are lots of conspiracy theory that its origin was from the Wuhan Institute of Virology. That is why when health officials they have visited in Wuhan Institute of Virology, they have tried to see all the documents, how the study is actually happening, the study design, the protocols, guidelines, SOPs, everything was evaluated very properly. So, if any mishap may happen at that time, your these things will be come to the picture. Then each dose level expressed in the milligram or per kilogram of the body weight and other appropriate unit of the test and control article should be to be administered, and the methods of frequency of the administration. These things also very much needed. Okay. How much dose you are actually giving in per kilogram weight or appropriate unit, okay, test or control article. So, such things also should be mentioned. The type and the frequency of the test, analysis and measurement to be made, the record to be maintained. You have to maintain all the records, that how you are performing the test and what are the steps, how you are actually following the flow chart of the test. Okay. And also the date of approval of the protocol by sponsor and the date signature of the study director. See, again study director actually coming in my previous lecture I have told you the role of the study director. So, the date of approval of the approval and protocol by the sponsor and the dated signature of the study director. The date from which your experiment is actually starting, it will actually create a very big role, because it will be helpful for the health inspector, if they are actually inspecting, the health inspector to identify or to understand how the progress was happened or how much progress actually happened with your study. For example, you have started your study in December 31st of a year. After that, you have faced some issues like power cut or transport related issues, or you have not received the animals, or you have some other issues. So, your activity was actually that is stopped for 3 months. In the next March or in the end of March, you have started your work. By the end of July, your work was not completely completed. Why? Because for 3 months there was lots of issues. So, by looking the sign of study director, the health officials or inspector, they will be able to understand that how much progress was happened, why it was hampered. In future studies, they will be able to encounter those issues. A statement of the proposed statistical method should be also used. See, statistical methods, it is very, very important, because this is the era of machine learning. I hope many of you are might be that is learning in the other subjects or other popular courses about the multiple linear regression, SLR. You also might have seen about various kinds of deep learning algorithm. So, why a statement of the proposed statistical method should be used, so that in future any of the data analytics scientists may work to see or to prevent the issues related to the delay of a study design, the delay of a success of a non-clinical studies. In that scenario, this data will be helpful for the machine learning scientist or data analytics scientist. They will be able to analyze by using this SLR, MLR and DL, they will tell that where the mistake has happened. Okay. So, now after learning about all those things, another thing I will tell you that all the changes or the revision of an approved protocol and the reasons thereof, therefore, shall be documented, signed by the study director and dated to maintained with the protocol. Now, let me tell you more to conduct a non-clinical laboratory studies. So, the non-clinical laboratory study shall conduct in accordance with the protocol. Obviously, you have seen all the details about our protocol, and then the test system shall monitored and conformity with the protocol, and also the specimen shall be identified by tag system, study, nature and the date of collection. This information shall be located on the specimen container or shall accompany with the specimen in a manner that avoid error in the recording of the storage data. Okay, see many of the cases you see there are three kinds of work. One is in silico, one is in vitro and one other is in vivo. In silico work, if you do some work in computer, if any mistake may happen, you can solve it by using the computer that is programs. In vitro, if any problem may happen, there are less chance to rectify this problem by using some manual method. And in vivo, if any problem or any issues may arise, there is I can say no chance you can rectify the issues. That is why you have to do your work very carefully. So, the information to be that is located in the specimen container or shall accompany with the specimen in a manner that avoid the error in the recording and storage of the data. Records of gross finding for a specimen for postmortem observation should be available to the pathologist when examining the specimen of the histopathologically. You see a pathologist when he or she will perform the histopathological study, there are chances he or she find some other observation, other results which you are not supposed to that is expecting. Why? You see there are almost for human there are almost 20,000 different protein coding genes are available, 20,000. For our drug discovery purposes, we are actually trying to target only one protein. That is why the famous quotation we got, one target, one drug, but if your drug may bind with other proteins, what will happen? It will stop some signaling process, it will stop some activities, and obviously with stopping some activities there will be some adverse reaction or some side effect. I can say there is some difference between adverse reaction and side effect, but you will be observing such kind of things. At that scenario, if you do some histopathology study, the pathologist will be able to identify that if any other kinds of cell signaling pathway was hampered or not. That is why it is very much important. So, the gross finding of a specimen from postmortem observation shall be available to pathologist when they examining the specimen in the histopathology. If your rat or if your specimen whatever on you actually working, if it is working very nicely, but still in histopathology you are getting a different finding, then you have to take care of it, because in future this will create a very big problem. How? For example, you got very good results and clinical trial etc., you are going very it has went very well, but after approval of the drug, when it will come to the market, there will be some post marketing surveillance. During post marketing surveillance, there is a very big genetic variation among our population. Those population having the over expression of such proteins, these proteins will bind with these drugs. So, the one protein whatever you are trying to target, your drug will bind with the other things. At that scenario, it will show the adverse reaction or side effect with a much more rapid manner, and then there will be some issue, and then your drug will get recalled. So, that is why you have to take care of all those things at the beginning, and that is why we are actually teaching you the quality assurance and quality. Okay. So, this thing should be available. All the data generated during the conduct of the non-clinical laboratory study, except those data generated by automated data collection system, shall be recorded directly, promptly and legibly with the ink. All the data entry shall be dated on the date of entry, signed and initiated by the person entering in the data. Okay. So, because why sign? You are signing just to validate date of entry on which date you are actually entering the data. Basically, you are actually trying our best to maintain the quality. If you just write the a table with a specimen and its results, this will not give you the proper answer, because in future if it is showing any adverse or other kinds of reaction, at that time people will ask, the health official will ask that why it is happening, show us the previous data. So, if all the data and everything is available, so at the scenario the progression, if they are actually doing some statistical methods, if they are putting all the things in the graph, then these things will be very much easier for them to understand. Okay. So, that is why in automated data collection, the individual responsible for the data direct data input shall be identified at the time of data input, and any change in the automated data entries shall be made so as not to obscure the original entry, and shall indicate the reasons of the change shall be dated, and the responsible individual shall be identified. If any human error may happen, that thing we may counter by this process. Okay. So, these are the things today I have tried to tell you about the protocol, where you have seen how protocol is actually playing a big role. And in conduct, let me tell you more about few things that I have not actually got time to cover. You see, protocol actually not only playing a big role in your this work, in your any of the studies of this kind of work whatever I mentioned, this protocol can be useful for other people also, who are trying to follow your this kinds of work. For example, somebody is working on cancer, and you know there are more than 200 different kinds of cancers are available. So, for your study one protocol worked very nicely, you got good results. Now, somebody working on a different cancer, you have worked on a prostate cancer, you have prepared the protocol and designed all these things very perfectly, you worked very well and you got very good results. Now, you are going to file some patent and get the approval, and you are going to marketing your drug. Somebody working not in prostate cancer, he is working in the lung cancer or other kinds of cancer. Okay. So, in that time also whatever the precautions you have taken, all of the things you have mentioned in your the protocol, how the data collection was happened, how you have seen the cell viability, MIT studies, also for example, cell apoptosis you have mentioned. So, these things will be helpful for other people to design their work in future. Also, for validating your work, protocol will play a very big role. And I have also told you about conduct of a non-clinical laboratory studies, how you can do, if it is a non-clinical laboratory, how you can ensure the quality, how you can maintain the data, how you can able to that is record the data, its input and other things. In my next lecture, I am going to tell you with a very detail about the reporting of a non-clinical laboratory results, how you can store and retrieve the record data, retention of the record, what is the disqualification of the testing facilities, ground of disqualification, and notice and opportunity for hearing of the proposed disqualification, and final order of the disqualification. These things I am going to tell you in next. After listening my next lecture, you will be able to understand, if you do, if you by mistakenly if you make some mistake during your studies, at that time how these things will be taken care of. The people those are actually working in this work, there are some cases your work might be get disqualified. So, that is it from my side. Thank you all of you for listening this lecture very patiently. I hope you got a very lots of insights. So, all the very best. We may see in our next lecture, and the next lecture will be the last lecture of our this particular unit. With this our unit will be also get completed. Thank you all of you. All the best.